And with that, I am going to pass this on to Dr. Mohamed Abdel-Rahman from Ohio State. So Dr. Abdel received his medical degree from Cairo University in Egypt and his PhD from The Ohio State University in Columbus, Ohio. He completed a general pathology residency at Theodore Ballars Research Institute in Egypt and also at the Menofia University in Egypt. And he's completed multiple fellowships, as you can see here. Liver Molecular Pathology Fellowship at the National Liver Institute in Egypt. Cancer Genetics Postdoctoral Fellowship at the University of Texas in Houston. Ophthalmic Pathology Fellowship at The Ohio State University, and also a Clinical Cancer Genetics Fellowship at OSU. He is currently the Associate Professor at the Department of Ophthalmology and the Division of Human Genetics. And his clinical interests include pathology, cancer genetics, and molecular genetics, and his research interests include hereditary predisposition to cancer, molecular genetics of cancer, particularly uveal melanoma and liver cancer. So we are very excited to have Dr. Abdel with us. I'm the expert in BAP1. And with that, I am going to hand it over to him. Dr. Abdel.
Thank you. Oh, can you hear me?
Yeah, we can hear you. Thank you so much for making time for us. We're really excited about this. And I'll be navigating the slides for you, so just tell me when to move next.
Okay, so thank Dr. Paul and Brenda for organizing this, and it's my pleasure to present to you today like what we know about the BAP1 tumor predisposition syndrome. Some of the slides that I will show today have been prepared by Rob Pilarski, who I've been working with for the last 10 years. Okay, next. I don't have any conflict of interest to disclose. Today, I will focus on the clinical aspects of BAP1, including the epidemiology, clinical spectrum, management guidelines, and current challenge and future direction. Next. So BAP1 or BRCA1 associated protein is it's a bona fide tumor suppressor. It's independent of BRCA1. Located on chromosome 3, which is deleted in multiple different cancer. It's a multifunctional debutynating enzyme with multiple different functions, including transcription, cell cycle regulation, DNA damage, which is very important, chromatin dynamic, and apoptosis. Most of these functions are in the nucleus, but there is also some cytoplasmic function, which is apoptosis. Because of the limited time, I would just mention a couple of things about the structure of BAP1. So BAP1 is moderate in size. It's 729 amino acid. What you will notice is in the N-terminus domain, you will see it's about 240 amino acid, and this is the DUB domain or ubiquitinase activity domain. And towards the carboxy terminus, in the last 10 amino acids of the protein, there is a nuclear localizing domain. Both two domains are very important for the tumor suppressor function of the BAP1. And some of the missense variants located in the DUB domain have been suggested as pathogenic. Also, the location of the nuclear localizing domain towards the carboxy terminus of the proteins indicates that almost all truncating mutation in BAP1 will be pathogenic. Next. So we really don't know the exact epidemiology of BAP1. The prevalence is still unknown, but what we know is the carrier frequency in non-cancer population is rare. So it's 1 in 27,000. This is based on the GenomeAd. In cancer patients, the largest cohort of unselected cancer patient was tumor atlas. And in that cohort, out of 10,000 cancer cases, there were about 8 patients with BAP1, including one with large deletion. In terms of general population, there have been a couple of population-based studies for renal cell cancer, suggesting BAP1 around 1%, mesothelioma ranging from 1 to 3, uveal melanoma about 1 to 2, cutaneous melanoma rare, like 1 in 1,000, and hepatocellular carcinoma, which was our surprise, about like 0.5%. But cholangiocarcinoma and meningioma also are there are like significant cases with germline BAP1. Next. I'm just including this from the paper, the TCGA paper on germline mutations in multiple different cancer genes to show the breakdown of the 10,000 patient in that cohort. And I want to point out like something like breast cancer here. There was about 1,100 patients. I don't know whether it's clear for you or not, but there are about 1,100 patients with breast cancer. Ovarian cancer, there are 400. So none of these patients actually had BAP1, suggesting that this are rare phenotype, if even like part of the phenotype of BAP1. Okay, next. In terms of public databases for ClinVar, currently there are about 1,000 germline variants in BAP1, and these are variants, not cases. Okay, so the cases is a little bit higher. One of the limitations of the ClinVar is that there is no phenotypic data, so what you will find is just like consistent with BAP1 tumor predisposition, but they don't list like what type of cancer in that particular patient. And I will talk a little bit about the big consortium in the next slide, but the overlap between the ClinVar and the big consortium is about only 10%. Currently in ClinVar, there are about 127 likely pathogenic variants, including one founder mutation, and there is about like 500 VUSs. One thing which you need to pay attention to is that there are at least 3 large deletions, and we are aware of 5 additional large deletions, including like whole gene deletion. So whenever you're doing testing, I mean, you need to make sure that the testing includes that. And I have been actually keeping tally of the number of cases since BAP1 have been introduced in the clinic. So this like diagram show basically how there is a steady increase of these cases in ClinVar. Next. Because of the limited number of publication on the clinical phenotype of BAP1, we established the big consortium about like a year and a half ago. And we were very inclusive. We reached out to every single group in the world who published anything on BAP1. This, of course, germline. And almost all of them actually contributed cases and like were part of the consortium. So now we have more than 150 collaborators from 19 different countries. But as you can see in just like this map, most of these cases are either from United States, Europe, especially France, Australia. So there is kind of like the number of patients with from white Caucasian background is dominant in the cohort. Next. We reported the first consortium finding close to a year ago, and that was on 141 families. This would include the 141 probands and about 183 carrier relative. And the summary of the consortium data suggested that there are 4 cancer: uveal melanoma, mesothelioma, cutaneous melanoma, and renal cancer, which are the 4 kind of big part of the clinical phenotype. Other cancer like meningioma and non-melanoma skin cancer, especially basal cell carcinoma, could be part of it. One thing which I also want you to notice in this table is that we assessed biallelic inactivation in many of these tumors. And in all except breast cancer, actually, the tumor from this individual actually had biallelic inactivation in BAP1. The other thing which I want you to notice is age. So you can see that the average age in this patient is much younger than the average age expected in general population. Next. So the summary of this consortium finding is that BAP1 is autosomal dominant with likely high penetrance. We haven't seen any genotype-phenotype correlation. Major syndrome feature, at least 4 cancer: uveal melanoma, cutaneous melanoma, mesothelioma, and renal cell carcinoma. But there are possible other cancers, especially basal cell carcinoma, meningioma, and cholangiocarcinoma. Next. And this is the first family that we reported, which actually it's an excellent or a typical example of a BAP1 family. What you see here in this family is the diverse clinical phenotype. So you'll see uveal melanoma, lung cancer in this proband. You will see uveal melanoma, another individual here, cutaneous melanoma, mesothelioma, meningioma. One thing which you can notice in this individual here, at the time of accrual, she didn't have any cancer, but unfortunately 2 years after accrual, she developed peritoneal mesothelioma, and she passed away from that. So almost every single middle-aged individual in the family with germline BAP1 actually developed like one type of cancer associated with the syndrome. Next. In the next few slides, I will talk a little bit about the main 4 cancers as well as skin lesions associated with the syndrome. So the first one is uveal melanoma. Uveal melanoma, you can find it in the literature as ocular melanoma or choroidal melanoma, and they originate from the choroidal melanocyte. They're totally different biologically and genetically from cutaneous melanoma. So they don't have the BRAF, RAS, or any of those. I mean, they have their unique genetic change. And they're quite rare, I mean, 6 per million individual. Next. So for BAP1 and uveal melanoma, in a couple of population-based studies from Australia, US, and Finland, the frequency is about 1 to 2%, and this is unselected. This will go up to 20% in familial uveal melanoma, where you have two or more uveal melanoma cases in the family. And it's also high in families or probands with young age of onset of diagnosis, like less than 30, and in those with other cancer associated with the BAP1. So those will be the feature in uveal melanoma patient where basically with kind of red flag for you. Somatic mutation in BAP1 is quite common. It's 40% or a little bit higher than that. And the majority of this patient actually will develop metastasis. So mutation in BAP1 is associated with an aggressive clinical phenotype. We don't have enough patient with germline mutation to kind of estimate the outcome, but based on the somatic, what we observe in somatic, we believe that patients with germline BAP1 mutation are like part of this aggressive phenotype, and they need to be managed based on that. Next. The next tumor is mesothelioma. Mesothelioma is also rare, but like 1 in 100,000, and it ranges from different state based on the asbestos kind of level in the environment. It primarily affects lung, but sorry, pleural, but you can also find it in peritoneum. And as you all know, it has strongly linked to asbestos exposure. Currently, there is no consensus on screening, and usually these patients are diagnosed late with poor survival. Next. For BAP1 and mesothelioma, if you have unselected mesothelioma patient, the frequency of BAP1 will range from 1 to 3%. I mean, I think based on what I have read, I believe it's more toward 1 rather than 3, because this paper here was a little bit, I believe, like selected, but so that's kind of an unselected cohort. This go up a little bit to 8% in patient with familiar pleural mesothelioma. One thing unique about mesothelioma is that patient with germline or somatic BAP1 actually have a better response to treatment. So this is contrary to what I just mentioned in uveal melanoma. And of course, there are some experimental work suggesting that like germline heterozygous BAP1 mutation predispose animals and probably subjects to increase asbestos sensitivity. Next. There have been like several studies, both in somatic and germline, showing that subject with BAP1 are actually have better prognosis. The germline study, which came up from Dr. Merking group, I believe last year, showed that or suggested that pleural but not peritoneal mesothelioma are associated with better outcome in patient with germline BAP1, and this was basically linked to response to chemotherapy. Next. Next cancer is renal cell carcinoma. Population frequency range from 1 to 1.5%, and this have been shown in like different cohort, including one from the US and one from China. There have been some evidence suggesting that BAP1 mutation are associated with more aggressive clinical phenotype, and this of course like in somatic. We don't have enough patients with germline to estimate their outcome. Another thing which I want to point out for renal carcinoma, it's the vast majority will be clear cell, but there have been reports of papillary and chromophobe. So just like I know this could be, especially in somatic, I mean, people have linked that to clear cell carcinoma, but other like morphology are also associated with germline BAP1. Next. The most challenging lesion in BAP1 will be the melanocytic lesion, and the challenge is multi-level. The first thing is naming or nomenclature. So you will find in the literature and in pathology report multiple different names for the same lesion. So some of them is atypical Spitz tumor, nevoid melanoma, this M-BAT, melanocytic BAP1 mutated atypical BAPoma. So that's kind of like will be your first challenge, is like, okay, like what's written is it that lesion or not? The one which have been now recommended based on the WHO classification is BAP1 inactivated melanocytic tumors. Okay, so that's kind of like the one which have been recommended. Next. The next challenge is the frequency in patient with BAP1. In one study where they assessed that very carefully, they estimated that this is one of the most penetrant lesion in BAP1 patient. Unfortunately, not every group actually assessed that, including ours. And the reason is that in order to diagnose this lesion, you need to biopsy them. And we don't believe that because they are benign, we don't believe that you need to biopsy every single individual. So that's kind of like the challenge with kind of estimating the frequency of this lesion. For For you, I mean, if you want to diagnose them, the most consistent thing is this lesion will have both BAP1 loss and BRAF mutation. So you look for either BAP1 loss by histology, by immunohistochemistry, or if they did like genetic testing, so they will have like a biallelic activation in BAP1, as well as the BRAF V600E mutation. So that would basically be consistent with this lesion. Next. This is just like the morphology. So you can, as you can see, it's a skin-colored papule, slightly elevated. They have, as I said, both BAP1 loss and BRAF mutation. And this could be like only like a single lesion, or next, or multiple lesion up to 50 or 100 in the same individual. So that's kind of it varied from one family and one individual to another. Next. Another challenge with BAP1 is cutaneous melanoma. The frequency in general population is quite rare. In one big study, they didn't find any pathogenic variant. In another study, they found 3 in 1,000. So we can estimate the frequency to be about like 1 in 1,000. In patient with family history, it doesn't really increase that much, 5 in like 1 in 200. And in our cohort, out of 53 patients with cutaneous melanoma or cutaneous and uveal melanoma with no other cancer. This is a key issue. No other cancer in this cohort. We didn't find any of them with BAP1. Okay. It was also rare in a cohort from like a decent cohort, 450, where they found was familial skin melanoma, where they found it only in 3 cases. So again, I mean, this is like raise a question why we see it more frequently in our patients, and whether like some of our patients diagnosed with cutaneous melanoma are indeed this melanocytic like benign lesion or not. So hopefully there will be like a review of some of this lesion, and will be coming out from one of our collaborators, who was a dermatopathologist. He has been collecting this lesion and this patient for a while. He promised that he will have this soon. So hopefully that review or study would answer some of this question. Okay, next. So based on what we know, we have proposed some testing recommendation, which would include a family member of germline, of a subject with germline BAP1, of course, or a patient with 2 or more of these tumors themselves, and or first or second degree relatives. The only exception would be cutaneous melanoma because of the data that I showed you. But our recommendation is that if you are planning to test a patient with cutaneous melanoma for like genetically, I mean, you better include like BAP1 in the panel. Next. And our proposed screening recommendation is focused on the 4 lesions: eye melanoma, mesothelioma, cutaneous lesions, and renal cell carcinoma. So what we currently recommend is for eye melanoma or uveal melanoma, we recommend starting screening at age 11, because youngest age so far have been 16, and we have actually observed more than 1 patient with around that age. So at least I believe 2 or 3 patients at 16. There was another one at 18. So there have been more than 1 case actually with such young age. What we recommend currently is they need to do like an annual dilated eye exam and at least a fundus photography. If they find any lesion, they need to refer them to ocular oncologists who are more specified in management of this lesion. If the patient is diagnosed with uveal melanoma, we recommend surveying them as a high risk for metastasis. For mesothelioma, we actually there is no like screening guidelines exist. So we recommend just like annual physical exam. We're a little bit caution about chest X-ray and CT because of the radiation. So kind of like we hold off on that. The only thing is if you are going to do like MRI for renal cancer, I mean, you would recommend evaluation of peritoneum and pleural if possible. For cutaneous melanoma and basal cell carcinoma and this atypical melanocytic lesion, we recommend an annual dermatological exam with full body exam and maybe like at least photography, and then follow up by a dermatologist. We don't currently recommend biopsying these atypical lesions, but it will be up to the dermatologist. So if there is a change in the color, shape, or anything suggesting that they are progressing, I mean, they would be biopsied. And for renal cell cancer, currently we recommend starting at age 30 with an annual abdominal ultrasound, and every other year doing MRI. So that's kind of our kind of screening guidelines. Next. So this would bring us actually to the challenge in BAP1 and future direction. So the first challenge is the cancer spectrum and the phenotype. We are kind of confident about the 4 cancers, but something like liver cancer and cholangiocarcinoma is a little bit like we still need more evidence for that, and of course that would be important for like the screening guidelines. Penetrance, it's also like the vast majority of the families that we have now are were enrolled based on like family history. So we probably are biased with overestimating the prevalence. So we need more population-based study. Management guidelines. We get like lots of inquiry about radiation, whether for diagnosis and treatment, and we really don't know like what to recommend for this folks. So we definitely need more information from like longitudinal cohorts in order to be able to get some evidence-based recommendation about that. And pathology review, I talked to you about the issues with melanocytic lesion and skin melanoma. So that will be like an issue to take care of. Also like frequency of large deletion and rearrangement. The vast majority of studies actually were done using Sanger. So probably we're underestimating frequency of large deletion. In our cohort, we estimated large deletion about like 10%, which was the same in the TCG cohort. It was also 1 in 8. So it's there, and you need to be aware of it. And if the testing is done by like Sanger or whatever, I mean, you need to reflex that. Okay, next. Modifier genes, gene-gene interaction. I mean, I showed you that the vast majority of family are from white Caucasian, so ancestry will be an issue. Also, asbestos exposure will be another issue. VUSs, I mean, this is really a big headache for now. I mean, we are receiving increasing number of them, and we don't have a good test to check their pathogenicity. So we are working on that. And finally, the targeted therapy. There have been lots of discussion about PARP inhibitor. At least there is, I believe, like 5 or 6 clinical trials where BAP1 is like one of the candidate genes, and they are both on all cancer as well as on mesothelioma, renal cell carcinoma. So hopefully we will know a little bit more about the efficiency of PARP inhibitor in this patient in the next few years. Next. So your role is will be very, very important to help us increase accrual for BAP1 patient. We have an ongoing study at Ohio State. We are accruing any patient with a clinical phenotype suggestive of BAP1, including uveal melanoma or any of the at least 4 cancer associated with the syndrome. We do like a research testing, and then we confirm in a CLIA lab if the patient is positive, and we can do counseling, or we can just like give the clinical testing result to you to counsel. We are also interested in any patient or family member who was BAP1 pathogenic or VUS, especially if the family history is consistent with the clinical phenotype, in order to be able to develop, better develop and test the functional assays that we have. And we're hoping that if we get some funding, we can establish a registry of BAP1 families. Next. I think I will stop here. I will just go to the last slide where my collaborator to thank them. Yeah, so I have been working with Rob for more than 10 years. Actually, with this group, I have been working for a very, very long time. Special thanks, of course, to Dr. Davidorf, Dr. Sabula, who are ocular oncologists, and Nick Hayward from Australia, who's a co-leader of the BAP1 consortium with me, as well as Dr. Juan Jeter, who used to be at Ohio State but moved to Utah just like last month. So with that, I would thank you very much for listening, and I will be happy to take any question.